bold new strategy to suppress HIV passes first test. A stripped-down HIV genome can interfere with the normal virus replication. More than 15 years ago, University of California San Francisco biophysicist Leor Weinberger proposed an audacious new strategy to beat the AIDS virus, giving already infected people an engineered version of HIV. The variant, stripped of nearly all its genes and designed not to cause disease, might outcompete and suppress the natural version, he thought. The therapeutic interfering particle, TIP, his bland term for the defanged HIV, could even spread person to person, reducing AIDS prevalence globally. This week brings proof of principle for the first part of Weinberger's vision. A single injection of TIPs into monkeys already infected with a simian version of HIV knocked down their virus levels 10,000-fold for prolonged periods, he and colleagues report today in Science. Plans are already underway to test this fight fire with fire approach in humans. If it proves safe and effective, Weinberger imagines it could be offered to everyone who learns they are infected with HIV. Many people living with the virus have difficulty accessing antiviral drugs or taking the pills every day, he notes. I think we need to try something new. This is an exciting paper, says Nobel laureate David Baltimore. He and his wife Alice Huang, both virologists at the California Institute of Technology, in the 1970s described similar disabled versions of poliovirus and vesicular stomatitis virus, which sickens livestock, that naturally emerge in cell cultures and interfere with the intact virus. We never put in the effort to make it a reality, says Baltimore, who is excited that now, there is a chance that the therapeutic potential of defective interfering particles will be realized. Weinberger became intrigued by the novel antiviral strategy in graduate school when he learned that cultured influenza virus would sometimes produce defective variants that blocked the wild-type virus. He began doing experiments with defective HIVs in 2007, but it took until 2018 to develop one with the properties he wanted. He stripped or crippled HIV's genes to create the TIP's RNA genome but left sequences that help it replicate. To do so, this neutered HIV genome parasitizes the molecules made by the HIV already infecting the cell, including its capsid proteins, which form a shell around the TIP's RNA. Because the TIP's genome is simpler than HIV's, the variant can copy itself more rapidly. Most of the interference occurs because the TIP genomic RNAs are grabbing capsid better because there's more of them, Weinberger says. The monkey study, done by a team led by Nancy Hagewood at Oregon Health Science University, injected Weinberger's TIPs into 6 infant macaques and 24 hours later infected them with a HIV, a lab-made virus that combines the simian immunodeficiency virus with the gene for HIV surface protein. After about 30 weeks, 5 of the 6 TIP-treated monkeys were healthy and their levels of HIV had dropped from a peak of about 100 million copies of virus per milliliter of blood to 10,000 copies per milliliter. 3 of the 4 control animals, in contrast, got so sick from HIV they had to be euthanized at 16 weeks. John Coffin, a retrovirologist at Tufts University, notes the HIV monkey model isn't really AIDS, as the animals develop disease far more quickly. But he thinks the strategy should be tried in people. It won't be curative, but it could reduce people to HIV levels where the progression of disease is unlikely, Coffin says. Antivirals for HIV have improved over the 17 years that Weinberger has spent refining TIPs. Recently, long-acting injectable versions of HIV drugs have proved to work for up to 6 months, potentially making it far easier for people to stick with treatment. But they're still antivirals and require lifetime treatment, Coffin says. The TIP idea, he says, is certainly the most interesting new approach for a long time. This is lovely, promising work, says Mary Carrington, a retrovirologist at the National Cancer Institute. But she says the risks of injecting people with neutered HIVs must be carefully explored. The TIPs might lead to dangerous inflammation or to immune responses that could defeat them, allowing HIV to resurge, although those problems weren't seen in the monkey experiments. The TIPs could also trigger cancers when their genomes insert themselves into human chromosome, but Weinberger notes that HIV itself could theoretically do so, and only one case is known. Like HIV, TIPs could be transmitted to others, primarily via sex. Some see that as ethically problematic. To Weinberger, it is a public health benefit that could help curb the HIV/AIDS epidemic, akin to the way the weakened virus in oral polio vaccines spreads to unvaccinated people, boosting their immunity, because many people now taking antiretroviral drugs might stop taking them in favor of a single shot with a tip. Weinberger plans to do more monkey studies to explore the effects of the switch. Discussions with the U.S. Food and Drug Administration about staging a human study have also moved far along. The volunteers would come from a cohort of people living with HIV who are controlling their infections with drugs, but have other illnesses that are terminal. The study would give them a tip and then stop their antiretrovirals to assess what happens to viral levels. After the volunteers die from their other illnesses, researchers would analyze their tissues to see whether any tip-related inflammation or other problems occurred. Weinberger hopes the study, which would be funded by an existing California state grant to test new HIV interventions, will begin early next year.